So far in this class, we've looked at how atoms can build up life's building blocks. And then we followed those building blocks through amino acids to protein machines to molecular assembly lines. And now we're going to back up a little bit and look at the building blocks for DNA and RNA and see how those fit together and talk a little bit more about DNA itself. DNA has been called life's blueprint. But a blueprint has a lot of information about how to build the house or the shopping center or whatever it is. And I don't like to think of DNA exactly like that. I think it's more like a menu, a list of choices that you can choose from. It tells a cell what it can order in terms of proteins and strands of RNA. And all the things that it orders has to then go through the ribosome if it's a protein to be made, like we talked about before. I don't even think of the DNA menu as a shopping list, like, okay, go get this, go get that, because it has everything that is allowed to be made in the cell. It's the entire catalog, the entire menu. So how does the cell know what to make and when to make it? Well, a cell uses a complex molecular program to decide what to order and when to order it. This program is in some way part of the DNA, but it's also on top of the DNA. If you put a bare strand of DNA into a liquid, it really wouldn't have all of the information it would need in order to execute that complex molecular program, because that molecular program involves proteins that are already present that can turn things on and turn things off when the cell needs certain things. So this is a molecular program, and it's very similar to a computer program. Instead of microchip elements that have logic gates and things like that in a computer, the cell has molecular logic elements that turn on and off. Now, the nice thing about these molecular logic elements is that they use fuzzy logic. So it's not just a 1 or a 0, an on or an off. It's sort of mostly on or partly on. And fuzzy logic is actually better for what needs to happen inside of the cell, because everything is being bounced around stochastically, it's called. So basically, they need to have this fuzzy logic in order to get anything done. So that's what's allowing the cell to do all the different things it does. And once the cell decides what to order, the DNA then does become more like a blueprint, because it tells exactly how to build that particular item that has been ordered off the menu. So in that case, it really is like a blueprint. DNA, as we talked about before, is built from 4 letters, A, T, G, and C. You can look at the images of those different rings and study their differences. RNA is built also from 4 letters, and instead of T, it has the letter U, which is actually really a lot like the letter T. They're very similar. The DNA synthesis assembly line looks incredible. Because it has to synthesize a strand going this way and a strand going that way, it's constantly pulling on and off pieces, helicases, and all of that stuff. So I think it looks like a spaceship, something from Star Trek or Star Wars. The mRNA, as we discussed before, is the messenger RNA. And it is kind of like a photocopy of the DNA. Just like in a very important library, you might have some really rare books, and the librarians in that library would be very resistant to you taking those and bringing them home with you. So that's kind of how it is in the DNA. The nucleus and the holes in the nucleus, the nuclear pores, are very carefully guarded, and DNA does not get to come out. Instead, what happens is, just like in a library, you might be allowed to make a photocopy of those rare and precious books and bring those photocopies home, or take a scanned copy on your phone. Similarly, you can imagine inside of the nucleus, the mRNA is a very temporary copy of the more precious DNA, and it can go in and out of the nucleus no problem. The mRNA synthesis responds to the molecular program that we just talked about. So it is being made when the molecular program and its fuzzy logic says this is now time for the mRNA for this little piece of DNA to be made. mRNA stops when it gets to a termination signal, which is also part of the molecular program. And that resulting mRNA is ready to be processed by the ribosome, but it often has to be spliced together. Some portions of the mRNA must be removed. The remaining portions need to be put together, fused back together. And sometimes pieces of that mRNA are rearranged back and forth. So there's interesting things going on. And as we talked about before, RNA can act as an enzyme, and some of the ribozymes are actually self-splicing strands of RNA. So they do that to themselves. mRNA is certainly not the only kind of RNA. There are pieces of RNA, as we talked about, inside of the ribosome. The business parts of the ribosome are all made of RNA, the parts that actually do the jobs. RNA is not just a photocopy of DNA. It's not just the same 4 letters of information and their particular order. RNA has more flexibility than DNA, so it can fold up into shapes kind of like protein is folded up into shapes. So that opens up a whole new range of possibilities that the original DNA didn't have. There's a special kind of RNA that's worth learning about called transfer RNA, that's tRNA, and it's folded up into a bent L shape. That small little L is what fits into the ribosome and connects the mRNA with its sequence of letters grouped in 3s, because each 3 codes 1 letter of protein. And so that's kind of the couple between the 3 letters of RNA on the mRNA strand and the 1 letter of amino acid on the growing protein strand. So tRNA transfers the signal from the messenger RNA into the growing protein strand. And they talk about the anticodon and the codon fitting together. Those 3 letters fit together. And the codon table, which is what we were talking about earlier, 1 in a million, or the best of all possible codon tables, is the sort of decoding ring that you would use to say, okay, these are the 3 letters that code that particular letter. So that all is going on inside of the ribosome all the time. And a DNA strand that was exactly like the tRNA would have a hard time folding into that L shape. So it again illustrates how RNA is more flexible. Now, people talk about how RNA is the beginning of life and DNA is the most important molecule. What about you? What do you think is the most important biomolecule inside the cell? Is it protein, which we learned about before? Is it DNA, which is the blueprint of life, or like as I like to call it, the menu of life? Or is it something else? So what do you think? Think about that. That's a little activity for you to understand the differences between the main 3 biomolecules, protein, RNA strands, DNA strands, and find out which one is the most important to you. Make an argument for that and tell people or tell yourself why you think it's the most. Now, a lot of people in the past have really put an emphasis on DNA and RNA. For example, Carl Sagan, who was one of my childhood heroes, said that everything you need to know on how to make a human being is in the DNA. What might be wrong with that statement? Is it an overstatement of the fact? The same guy, Carl Sagan, speculated that the first life on Earth was an RNA molecule. Now, how much RNA do you think he meant by the first life on Earth? Was it 1 strand? Was it a whole bunch of strands? Was it a whole bunch of strands inside of something? And if it was like that, then what does it mean to say that life was RNA, or the first life on Earth was an RNA molecule? So those are things that, back when he said that in the 1980s, may have not been totally clear to him, but as we've learned more, it has become more clear what it takes to make the first life form. Now, getting the first strand of RNA itself requires RNA to self-assemble. And everything we've learned so far in this class is about how molecular machines assemble the building blocks. But how would that happen on its own? Would there be a way for the letters of RNA to just come together on their own in a certain pattern that could then fold up into a particular shape? What do you think would take to assemble RNA on its own? And do you think that there would be enough RNA and enough time on Earth or on all the planets in the universe for that to happen? That's a good thought experiment to think about for yourself. Well, we're done with learning about the RNA and the DNA, and hopefully you've understood how they are fit together and the flexibility of RNA and the importance of DNA. And this kind of summarizes the going from the building blocks, the 4 building blocks of those biomolecules up to the more full molecule. If you're digging a little bit deeper, you may want to read the textbook. There's several leafs on how nucleic acids are put together, how DNA is replicated, how RNA is different from DNA. And a lot of these leafs that you should read for this lecture are reviewed. So I'd like you to focus on the machinery that builds DNA and RNA so you can learn exactly how a helicase has to split apart the DNA and how the RNA is formed, etc. So good luck reading all that stuff.